Today we're going to talk about DNA replication, more specifically. That is, how does DNA copy itself? And this is critical because if we remember, every single cell in our body contains a copy of our DNA, and contains a full set of our DNA. So anytime we make a new cell, we have to make copies of our DNA for that new cell. And then when we go to make children as well, we need to copy our DNA to go ahead and pass on to them, so they can have some of our traits. So we're going to talk about how we go from one molecule of DNA to having two molecules of DNA. First thing we just have to review, and this is from our lesson on DNA structure, is that first of all, that DNA strands run from what we call 5' to 3'. And if you remember, I said this has to do with the number of carbons that are on this sugar molecule, deoxyribose. So at the top of the sugar molecule, you have the fifth carbon, and at the bottom of the sugar molecule, you have the third carbon. But the scientists just use this as a way to orient or know which direction their DNA strand is running in. Which direction is the sugar facing? They call it 5', 3'. So it's just a way to tell direction. And that the strands run from 5 to 3, and they run in opposite directions. So if you look at this double-stranded DNA molecule, it has two polymers, but one polymer runs from 5 on the top to 3 on the bottom, whereas the other strand runs from 5 on the bottom to 3 on the top. So they run in opposite directions. And this is just important to remember because it has a lot to do with how DNA replicates itself. The other thing I want to just quickly review is the cell cycle. So we talked a lot about the cell cycle, the life cycle of a cell. Specifically, what we're talking about today is this S phase of the cell cycle, or DNA synthesis. So if we remember, during the S phase, that's when DNA becomes uncoiled, and it becomes replicated for the new cell in mitosis. So again, we're turning one cell into two. That new cell we make needs a copy of the DNA. So we're focusing on this S phase of the cell cycle. Well, how do we go ahead and make a new copy of our DNA for that new cell? So it has a lot to do with making these new cells. So let's take a look at a DNA molecule, and I have a short strand of DNA here, and you can see that the double-stranded DNA molecule here, it has that complementary base pairing. So we have A on one side, we have T on the other. If we have G on one side, we have C on the other. If we have C on one side, G on the other, et cetera, et cetera. So the first step in making a copy of my DNA, and this is pretty interesting, is because our DNA is two-stranded, we can use each strand as a template to rebuild another strand. So the first step to making a copy of our DNA is simply to split these two strands up, or split these two polymers of the DNA strand up. So to do that, the cell has a special chemical enzyme called DNA helicase. And you can think of this kind of like the zipper pull on your sweater. Basically, it just runs down through the DNA molecule, and it breaks the chemical bonds between each pairs of these bases, essentially unzipping the DNA molecule. Now, what's going to happen from here is as that DNA helicase goes through the DNA molecule, following behind it is going to be another enzyme called DNA polymerase. Basically, what this enzyme does is it looks and it sees, well, what base is on this strand right here. It says it's a G. So I'm just going to attach the complementary one to it, a C. And this is where that base pairing comes in handy. We said that a C will only ever pair with a G, a T only with an A. And you can think of it, and I've kind of drawn these shapes here like puzzle pieces, right? So the C can only fit with the G, so polymerase knows to attach that complementary base. And if you think about this, if I do this, I'm building the exact same strand I used to have attached to it. I'm just, looking at what base I have and attaching the complementary one. So this is building us a new strand of DNA. However, replication is a little bit complicated. DNA polymerase, this enzyme, can only work in one direction. So remember I said that DNA is run anti-parallel. It runs in opposite directions. So DNA polymerase can only build in what we call the 5 to 3 direction, 5' to 3 direction. So you can see here by this arrow, DNA polymerase on this strand is running from this side to this side. It's following right behind the zipper. and it's going to go ahead and make a copy of that DNA. We call this the leading strand. But however, something else happens on the opposite strand. So remember that I just said this DNA polymerase can only run from 5 to 3. So that means on this other strand, well, 5 is on this side and 3 is on this side. It's running in the opposite direction, like highway. So DNA polymerase on this strand can only build in the opposite direction, working backwards, right? So it's still matching up those bases, but it's doing them in the other direction. Now, you might see the issue with this. If the helicase, this enzyme, is unzipping in this direction, on this strand, the polymerase enzyme can just follow right behind it and build the new strand. However, on this top strand, it needs to work backwards. So it needs to start here and work backwards. And then what happens? It needs to start here and work backwards. So DNA polymerase on the other strand is constantly playing catch up. We call this the lagging strand. So this one is being built continuously, nonstop. This one has to be built in a bunch of short pieces, and you can see them here, a little piece here, a little piece here, a little piece here. We call these little pieces Okazaki fragments after the name of the person who discovered them. So you can see here we go, we're building. This one is constantly playing catch up. So it'll make a new fragment here, wait to unzip, make a new fragment here, wait to unzip. It's going to keep playing catch up. It's going to keep making these little pieces of DNA. Now, this is still building a second strand. It's just building it in a bunch of little pieces. Well, pieces of DNA are no good. So there's one more special enzyme, one more special chemical that our cells have called DNA ligase. And ligase is just responsible for joining these strands together. So that DNA ligase enzyme is going to go ahead and join those strands together wherever there's a split. And that way, this lagging strand can still be built into one continuous kind of piece of DNA or strand of DNA. And you can see here that through this process, just unzipping and building based on the complementary strand, you end up with 1, 2 strands of DNA. Now, what's important to note about these 2 new strands of DNA is that each one utilizes half of the molecule, or one polymer, from the original piece of DNA. So you can see here, these 2 in dark blue, these are strands from these are polymers from my original strand of DNA, and these 2 are new strands that are making up the other half of these new DNA polymers. So we call this semiconservative replication, and then each new DNA molecule is made of half of the old DNA molecule. And you can kind of see it here as I zoom out. You can see my original strand over here gets unzipped, and then my new strands are built of half of the original strand. So this is how DNA replication works. We just unzip, we build. What's important to note is that we're building backwards on this opposite strand.